Khalid? Okay, so maybe we can continue. Okay.
Okay. So first we introduce like how epichaperone formed in the cell. So basically, chaperones are involved in many functions, especially putting, folding, and other stuff. But due to in stress condition, these chaperones are make a like a multimeric complex that is called epichaperone. So in epichaperone, like the function of other chaperone, co-chaperone, other proteins are just trapped and just they are become malfunctioning. So that's why that disease is created. So in our lab, we have developed like some chemical probes, which are like PU beads and YK beads, which are specific for some chaperone member, specifically if you're talking about like HSP90 and HSP70, which bind with HSP90 and HSP70 as a treatment when we pull down the epichaperone from the sample. So once our chemical probe is just bind with HSP90, just we have like experimentally we have pulled down these all the epichaperone from the cell, and then we go for further quantification of these proteins, that what are the proteins involved in that epichaperone by using like mass spectroscopy and some computational pipeline. So here is like in that network, which is like we call that epichaperone that is involved in uh, our chaperones. Like till date, like approx we have listed approx 350 chaperone co-chaperones are known. So we have like just focus on specifically HSP90 and 70, and like nowadays we are focusing on also like CD37 and HOP chaperones, which are also like significant role play in the disease. So this is the simple pipeline which I followed for data analysis. In like once we have received the raw file, which is basically like a protein intensity file, and just we have processed. So for before processing the data, we have to remove some falses, including potential contaminants. There are many contaminants are involved during experimental work, so we have to identify it and remove. And there is a many reverse hits, like that is decoy protein, which is like pseudo protein, which is also need to be removed. And other protein, like many proteins are also uh, uh, peptides of many protein, which is identified by modification site, like mostly phosphorylation site. We also need to be removed from the main data. So once we have removed all the false hit, then we go for missing value imputation, because in the data there are too much missing values. So missing values generally came from like uh, two types, like missing at random, which is basically just like a fault of detector, like our machine failure. That's why sometimes the machine doesn't uh, detect the spectra. So that's why they have put the 0 and NA. But in sometimes like missing are not random. That's in that case the uh, missing values are biologically relevant. So like in a many sample that particular proteins intensity not shown. That's why there is a NA values. So we have to put some statistical value along with these valid values. So we use different method. Basically nowadays I'm using minded and normal distribution, which is give the good enough like real values as compared to other methods. There is another method like KNN and PPC, BPC, and other. So nowadays I'm using minded basically. Then once we have like fill the missing value imputation, then we go for uh, like identify differentially connectivity protein, change that all the data into log2 form, and calculate the fold chain t-test. And on the basis of fold chain and p-values, we have just identified some differentially connected proteins. Once we have proteins, here one step is missing for protein-protein interaction network, which is I'm trying to uh, explain in our some special classes. So I have not mentioned here. So once we have all the specific protein, then we go for pathway enrichment analysis, including we have make a clustering and all the data which is from disease versus like normal data, and then go for pathway enrichment analysis. Basically we here use GCA, gene set enrichment analysis, as well as we are using Fisher exact test. These are the two methods for pathway enrichment method. So we generally use both to validate the results, like predicted pathway predicted by GSA is enough is like validated with Fisher exact test. If we found like common function pathways are predicted from both rule, then we go for like these are the significant of particular set of proteins. Then once we have identified the pathways, then we go for like visualize. So we have like a reactome map of like approx 2000 pathways. So we have mapped our predicted pathways over it. So that is a very good visualization of pathway in a one graph. So this is the overall our work nowadays. So this is the like a simple file format of raw data, which is started with like majority protein IDs, protein names, and we are working on intensity data, elective intensity, and yet total number of peptide spectrum count. So for analysis, this raw file we use basically a Perseus MaxQuant tool, which is analyze this data. So here, if you ready, so I can start the Perseus tool to analyze the data. Okay. Because in next slide, because this is a protein induction, which I will try to explain in next meeting. So here I will try to finish the protein processing part.
Okay.
So I'm sharing like a Perseus. So if you also like download it, so I can share the raw file. You also can try and join.
Yeah, that's what I wanted to ask you. Can you share the data
sharing.
file, because we already installed it?
Okay, please. Yeah, I shared.
Okay, I downloaded it.
Okay.
Thanks.
Download. This Perseus software is not for specific for proteomics, but we can use in many ways like transcriptome data and GS data and many other very useful tool.
Okay.
Can I start? Yeah.
Yes, I think
Okay.
so.
So first we have to select our raw data file. So here that is the one option like generic matrix upload, which is our raw data file. So first we have to upload, like this window is open. So first we have to go and select our file. like this is taken like text file as well as CSV file accepted. So we have like CSV file. So just we go. So there is like too much data. All the data in a column file are shown here. So first, like we have to choose what type of analysis we want. Like if you want intensity data, so you can take only intensity. If you want to LFQ intensity, so you can take LFQ intensity. So what are the different intensity and LFQ intensity? LFQ intensity are already normalized intensity, and intensity only intensity is the like overall intensity captured by our detector and just on. So most of the like expert recommended us to use LFQ intensity. So I'm using here LFQ intensity. Like select LFQ intensity and shift here. So and just insert. So in the main column, all the LFQ intensity are there. So here other information is also there. We can most of the information is just not for us, so we can remove it also. So, but here I will keep all the information. Just go and okay. So here we have all like we have the matrix of our raw data. Okay, everyone has.
Yes.
Okay, so like here, like how many like 4,254 proteins are exist in our file. So now we have to filter the data. So first we have to go to filter rows. Third one, filter rows, and go for categorical column. Select the category column. So here is the these authors. So first we start with identified by site. So the plus sign means there is a contaminants are present in our data set. So we have to just select only identified by site, and mode and filter mode remain same as the default, like remove matching rows and reduce the matrix. So like if here is 4,254 proteins are there, when you reduce the matrix or remove the contaminants, the matrix is like reduce. So like we start like this one. Now for all the contaminants which are identified by site peptides are removed from our matrix. So now we have only in the first raw matrix we have 4,254. After removal of like we have only 4,160. So again go in the same filter rows and category column and select reverse. This is the decoy sequence. Decoy sequence are also present in our data set. So we have to also remove use the same settings like this one. So matrix is again reduced from 4,168 to 4,128. So again we have to select third contaminant type, which is potential contaminants. Basically, these are keratin, BCA, some digestive enzymes, all there. So again we have to remove this contaminant. Okay. So we have removed all the contaminants, and now we have final 4,017 proteins in the list. So now next step is just go and log transform the data. So here these in the right column, these are the main column which need to be log transform. So this is the
Mhm.
X is the right column. Now we need to go to log transform all the intensity data among these columns. So these are now log transform, and NaN is just like a missing value, which I mentioned. So and now go to for like annotation rows to categorize the which are the wild type, which are the disease type.
Sorry, just a second. How did you transform the data into log?
Yeah,
Where
like
did you?
go to basic.
Basic. Okay,
Yeah.
okay. Sorry, I skipped. Okay, thanks.
So now again go to annotation rows in the second like in the fourth option in the lower rows category column. So like here is like progeria. So we have to select and press shift and like that. So we have a progeria dataset in a one group, and just untick this one values and just type pro on it. Yeah, any other name you want. So this is like identification. That is a basically this is the manifest for our sample data. So now select wild type, untick WT. Okay. Yeah. So now this is we have identified what are the our progeria dataset and what are the wild type in the. Now if you want to show like visualize our data sets First to go to like histogram according to intensity value. Like these are the columns we used. So just click on a visualization and histogram, just and this histogram is just like that. So here you can set how much column you need, 2, 3 in a one row, like that. So all these intensity values are plotted in a histogram. Now next step is just imputation. So here is us in a second row, third column, imputation. So there are many options. So first is like replace missing value by normal distribution. So like Max Quant team always suggests that when using imputation, try to use normal distribution. But many other author in the paper just suggest different methods. So it's up to you how much missing values data is real to your valid data. So here I will replace with the missing value with normal distribution. Just select and click this one. All the missing value was imputed. So this is the width and downshift. This is some mathematical terms where you can read that in a quotation. So I use basically default one, and in a downshift, that is the amount by which the distribution used for random numbers is shifted downward. This is the unit of standard deviation of the valid data. So we use default setting. So now all the missing values are imputed. Now again we can plot histogram to check the how. So So here is a selection from tab option. So we want to see where is imputation occurred. So you can see that in the orange color, these are the imputation occurs. So from this histogram, we can see that the imputation is just overlap as a real value. So we can say that imputation is done. So not like go over it. So like over one. So
Sorry, once again, can you I also missed this. Can you show me how you can overlap the two histograms, the one with the imputed versus just this last step?
like this is our matrix before imputation.
Okay, so
yeah.
Yes.
Good.
Once we imputed, so like now here we imputed. So go to visualization again, same.
Okay.
And make a histogram. Once the histogram is generated,
Okay.
just go to in the setting. Here is the setting. You see?
Oh, okay.
Selection from imputation. So once you just select these are highlighted. There is imputation done.
Okay. Good. That's nice.
So
Thanks.
once you imputation done, then we want to check the differential connectivity protein. What are the significant protein? So here is option in clustering section. Yeah, in visualization, first we check the correlation of different samples. So we can go and make a scatter plot. So column, because we need to column all these our data, progeria and wild type is in a column. So we select column. Now we scatter plot is created. So again, I'm repeating, like first go to visualization and go to scatter plot for seeing the correlation between among the samples. Once we created, these are the correlation. So here we can check the sample wise one by one. Like we are choosing same sample like that. So correlation, how much correlate? If one is correlate, that means a very good correlation. So basically we select Pearson correlation coefficient. So I'm again repeating, like first check the from here x and y. Check the sample wise correlation, and then go to the third one, correlation category, like point categories and correlation, and select the Pearson correlation. And other also many mathematical statistical calculation are there. So if you are interested, you can check by selecting any of them. So here I'm selecting Pearson correlation and just select this one option. So there is a very good correlation between these LFQ intensity and PRO3, like 0.93. So we can select any of us. And also, if you want to see what are the proteins, just go and select this one. Make a color, any color, and rename it. What you can check what are the proteins in our like a higher level intensity. So same thing we can, like if you want to make a scatter plot in a one figure, so we use like multi-scattered plot. So here is the option is a multi-scatter plot also, like a single one, like a multi-scatter plot. So here I just choose multi-scatter plot by using same column. So yeah, so these are the multi-scatter plot. Also here we can set the Pearson correlation. You can see and you can easily compare sample-wise correlation. One good thing that if you make some mistake, just select and delete here like this, remove selector.
Okay, and can you save this in a larger file or something like this?
Yeah, you can save like this one, export images.
Oh, okay.
And also you can save the session also. So if you like,
okay.
Yeah, so it is a good. You can also save the matrix, like right click and plain matrix export.
Oh, good. That's nice as well.
Any step you can save. It is better to like save session, so anytime you can just.
Yeah, sure.
If you want to delete something, just go select and just remove this one. So, so here you can't delete like in between, like you have to delete by lower one, like in a hierarchical manner. So you have to delete first lower, then upper, like that.
Okay, and if you want to perform some other operations, for instance, from matrix 3,
yeah, just
when you create, so let's say we are at matrix 3 and we want to filter by something else.
Yeah.
Will all these paths below it be destroyed?
This path is start in next path. This path is remain same, and it start with like I started with like something like I can log 10.
Okay.
Please,
oh, so it.
Okay.
Okay, good. That's nice.
Yes.
Okay. Nice, thanks.
Now, now we want to just go to make a volcano plot. Just there is option in third one column. So we make a volcano plot. Just click, select first matrix. Which matrix you want to make a volcano plot? So we have this matrix, and go to like volcano plot. So this is the progeria versus wild type. So you can select FDR value. Just click. These are the volcano plot created. So these are the minus log, negative log 10, log p values, and here we can see the also one matrix more. Yeah, you see the matrix. Yeah. Like all these, the plus sign symbols are just a significant proteins in our data set. These are the complete matrix. You also can save this matrix by clicking right click. So here is option. Okay, if you want to check what are the proteins in the in down regulated, so just select this one. You can check this, and you also select like you can set and select the bring selection to top. So all these selections are in the top, and you make also different colors. You can any colors.
I have a more little bit technical question. Sorry for interrupting. Now, what if we wanted to include our own transform? So let's say we have written in R or in something or in C a transformation of data that would be useful to be integrated in here. Is there something like introduce a custom transform or something?
Yeah. Like for imputation, there is a one more option. Like if you use R program, like this is there's a method.
Exactly like that.
Yeah. So here you can select your R script.
Okay, that's good.
These are the another method.
Okay.
So what is in the Perseus bin file, we have to provide the R file.
Okay.
R script. Then we select.
Okay.
So for this selection, you have to open R platform in a different window. Like here, I already open R platform. So that's why it looks green. Otherwise, it looks.
Okay. Good.
So you can use our customized script also here.
That's great. Okay, good. Nice.
So here, same thing we can identify. Once we now we have like a list of significant proteins which are like up high abundance and low abundance. So like if now our work is started, just take all the proteins from the matrix and make a protein-protein interaction network and highlight these significant proteins by different colors as I shown in here in my PPT. Some nodes are green color and some nodes are in red color. So many things we can do here, but like for us, these things are sufficient to get the matrix of significant proteins. So in proteomics, basically I did the same exact same thing for our data analysis. For next step, we can go for protein-protein interaction network. Here in Perseus, some questions please ask. It is better to if you run one by own, so more clear. One more thing I need to tell you, like in our data set, many proteins are like have like if you suppose here 8 sample in our matrix here. So sometimes what happen like all the 6 sample or 7 sample have missing values. Only one sample has a missing has a valid value. So this is like some kind of error. So we have to remove that column that rows all too from the matrix, because sometimes we can't say that only one column is detected and other column is just 0. So if you impute it, then that is a bias, tomb bias. Suppose here in the first column all these are 0, only 1 or 2 is like have valid values, but not all these not detected in the detector. So so many authors have suggested that it is better to remove, otherwise these proteins will create a biasness in the data selection, because as per imputation method these values already filled will be filled by some speculative method, but that is not real. Because if like 2 or 3 missing values there, then it is okay, because it takes like average and some other like in a minded, what in minded it takes it arrange the value of sample by decreasing order and take one quantile of the lowest values. So it is logically correct that it takes from the sample, it fill the values from its own samples. But if you have 1 or 2 sample, then it fill the missing value, that is some biasness. So we have to remove also those column also. So here is option in the filter rows in starting point. The filter rows based on valid values. So just select. Here is the option. So like a minimum number of values. Like in our sample, if 3 numbers of 3 columns have valid values, then keep it. Otherwise, discard it. So here is 2 options. Like in percentage. So like 70% values should be valid. Otherwise, discard the rows. So we can select here. So first we have to know that how much valid values are in our data, then select our option here. Like some, if you know that most of the values are like more than 70% are valid value, 80% are valid values, then select here.
I have a question here. Please. What if the missing data, so let's say you have 8 samples and you have missing data for all the wild type values. Yes. Yeah. And you have data for all the progeria data. Do you account for this case, or what do you do? In this case, we are working like we use basically hybrid, some like in a hybrid data, because it may be possible that in a disease sample the expression is proteins are expressed, but in the wild type proteins are not expressed. So this is also like a imputation challenge nowadays. Yeah, still, I know. Because in our AD data, so we have used male and female data. So what happened in the data sample, we found that some proteins are expressed in a male data, but not in a female data. If we imputed them, then we lost the significance of
we can't say that in the Alzheimer data these proteins are overexpressed in male sample but not in. So this is also a challenge. So with Dr. GC we are working. So for this basically I take hybrid approach. So like that sample if like 50-50, then we took and put the 0 instead of putting any imputation value.
Okay, so just a small value. So
yeah, we put only 0 there.
Okay. Okay. Good.
So this is the method how to remove these invalid columns from the rows. So here also you can select each group or like in group wise we can select. Like in a 8 sample, if in a 4.
Okay, so you can do this. Yeah, that was
like we can select like if a 2 column is a valid value, then reject.
Okay, good.
So this is the overall basic introduction of Perseus. So any questions from this?
So
no, but it's a great software. I really
yeah, it's great software, and we can analyze transcriptome data, ingest data too from this software.
Yeah, so it incorporates a lot of stuff that we did for pre-processing the data for imputation, because we were working on imputation, and all of this can be done just by simple clicks, and then we can just get the data that we want for testing the imputation. And and even if we can integrate then our own imputation method, it's really easy. So great stuff here. Thanks for showing it.
So next meeting I will explain protein interaction network, I think. So with
okay.
Okay.
Yes.
I will share some videos link for Perseus. So you Kalin and Noad, we can see and learn more about that.
Okay, thank you. And
thank you.
Yes. And can you share the slides from the beginning, so the PDF of the slides, please?
Yeah, I will share.
Okay. And for the next week, for protein interaction, will be the same Perseus soft or?
No, from different approach. Like we use different databases and like this. So
okay.
So we will like go one by one databases and just explain how to download the whole data set from the internet and then how to filter the data set for our use. Like that.
Okay. Yeah.
Next presentation. So I will more optimize over my PPT. Then I will share from next week in protein interaction network.
Okay. Yes, very nice. Thank you.
Thank you very much, Atam.
Thank you so much.
Okay. Thank you. Okay. Yes, it's very interesting.
Yeah, really interesting, and yeah, it eases our life a lot. So
yes.
Thanks.
Okay, thank you.
And if you want to,
if you even have a question, anytime you can send me a message. I will try to solve anytime.
Okay. Yes, maybe the next step will maybe to.
Yeah, in between in the days you can ask me. No need for formalities like meeting, and just ask me.
Okay. This is very useful.
Let's see you next week. Bye.
Okay, so thank you very much.
Thank you.
Okay.
Have a good day.
Good day. Have a nice day. Thank you very much.
Bye.